Okay, let's start off the class today. If you can hear me, always we check the whether you can hear my voice or not. Please raise your hand if you can hear me. Okay, so this is working. So good. So where are we? Oh no, shit. This one. Cool. Okay, so let's start off the by the review of our assignment. So you had the first written assignment last Monday, and I mean, as an overall, the performance from you guys quite, I mean, good. That's my first impression, and I think you got the scores, and I think most of the students get really good scores from the assignments. The different things compared to previous years, students well understand the sort of the basic concepts from the paper. So that's, I mean, positive thing. And I guess the probably the reason is most of students get the, you know, the 2 sets of the exams, and that's probably well prepared for the, you know, your background. So I think that's quite good. And then the writing itself is pretty good. I'm quite satisfied compared to previous years. So I mean, especially compared to last year's, I mean, this year's students, you guys done writing better than last year. So I mean, that's also another positive things. But some students are not really familiar with the, you know, writing in English, because you know, of course, you never really wrote the sort of this length of essay in your you know, lifetime. So probably some students felt awkward in writing. So I mean, I can see this, but I mean, at least you guys try you know, your best way. So that's a good thing. As I mentioned, that I mean, most of students get, you know, they get the sense from each question. So that's good. Describing the sort of experimental design, well done. I mean, this is for the finding de novo mutation and related to the disease. We study autism and in the paper, and then you guys get the sense of the de novo mutation from these exome studies, and also the describing the findings from the study, mainly for the, you know, the enrichment of protein truncate mutation and missense mutation in autism cases. And that's also well, you know, described by you guys, because you understand about the variant consequence, and also, you know, how to relate it, that issue to your reading. So that's good. And also, I mean, did quite good summary for your understanding as to the limitation of the study, and also the further direction from the study, because, you know, this is not going to be the end of the story. This is the, you know, the middle of the, you know, the discovery of many things in autism and genetic studies as well. And also, if you felt comfortable with this sort of understanding the concept, then you can apply this concept to other studies. Then you can easily understand the basic, you know, notion of the why we study large-scale genomics in the field, right? So these sets of questions for your learning, but also for your career as well. And also, why I'd like to, you know, give you the written assignment is that, you know, of course, you know, I can check up your attendance and, you know, 2 sets of question, mid and final exam, and give you grade, and that would be probably the easiest way to evaluate your performance, and probably the easiest way for both, right? Me and you guys, right? But having a the comprehensive evaluation during your university time, I mean, makes you, I mean, the better ability in a science career. So, and one of the aspects, I mean, definitely the writing, right? So writing or presentation is part of your sort of building your capacity in a scientific career. So this is one of way you can building your talent. So that's why I give you the written assignment. And also another aspect is that reading textbook is not really the comprehensive understanding. I mean, in terms of the latest science discovery. So I mean, reading the latest research from the the good journals like Nature or Science, that's definitely give you how to interpret the data and how to get the sense of the conclusion from each study. So this is the way we train in the session. And then one common feedback for some students, not all, is some of students are having quite high level of plagiarism, which is you might done intendedly or unintendedly, right? Copy and paste the text from the the paper to your assignment is not writing. This is definitely I mean just this is the part of the plagiarism. So you have to digest the sentence from the paper and put it into your writing, right? So this is building your sentence, not just copying someone else's idea into your content, right? So please be keep in mind that writing sentence by yourself, and also think about the level of plagiarism in your writing. So when you submit your assignment into Blackboard, you can get the scores from the safe per sign something, right? So this is checking your writing the plagiarism level, right? So this is, I think this is based on Turnitin or whatever Blackboard native system, but that definitely check your plagiarism. So some students, not some, few students, like 3 or 4, they did really great writing, and I can get that you guys well understand the concept, but your sentence is just a copy and paste. So so those students get like safe assignment scores like 70 or 80 something. So I mean, I wouldn't recommend next time. So next time will be penalty on that. So keep in mind that. And another few students, they did literally copy and paste. So almost 99% of the safe assignment. I mean, that's definitely surprising. I mean, sorry, I sometimes laugh. I mean, when I didn't really understand the situation. So so please keep in mind that if you write something, that has to be your sentence, right? So if you see the 99% safe science course, there's something wrong, right? So, but this time I really, put only one sentence on this. So I just 50% reduction from the penalty. So, but next time that's not allowed, right? So, please be cautious on that. Other than that, yeah, I mean, most students, they done really well. So, and this is the, Kaplani's study. This is one of the milestone paper in an exome sequencing, not just for the developmental delay. This is, I mean, including other large scale exomic studies. Kaplani's 2020 is one of the, the landmark studies. So I hope you really enjoyed that session for your writing. And I would say, after your graduation, after university time, you probably get like, multiple of the reading papers and summarizing into your writing, not just happening in, graduate, schools or research area. I mean, if you go to company, if you go to, I mean, other sectors of company, I mean, writing is the, daily life, right? So, I mean, this is not just for the, whatever, if you do research or something. I mean, this is not just a research area thing. This is just the basic things in a professional area. So I mean, if you go to media like Korean news, you see lots of reporters, they don't have any idea on how to write, how to read the paper, because they don't really get educated. So I mean, this is really serious problem. If you read the newspapers about vaccines or whatever the side effect in vaccine COVID-19, I mean, the reporter doesn't know anything about the science or the vaccine mechanism. So I mean, they write whatever they want, right? So I mean, they like clickbait the style reporters. I mean, this is the serious issue, and I mean, we are in a different position, and I mean, we also have the obligation to change sort of this situation in your in your time. So please keep in mind that the writing, reading is sort of part of your training, and also makes you better person in your next career. Okay, so this is the written assignment 1, and also the second assignment that's submit by 14th June. I mean, if you feel I mean, not enough time for the paper, I mean, we can extend the deadline for that. I mean, I temporarily put the deadline on the 14th June, but I mean, we can extend like a week after that. So see the schedule, and I probably do poll for you because you probably have lots of exams around that time. So, but anyway, preparing your reading in advance and also writing as well, and that will probably makes you much better in your exam preparation. And this time, another good study by I don't know this person, but this is really good paper as well, and also the same sort of sets of questions. So I hope you enjoy reading this article as well. Okay, so back to the previous session, in session 4 and 1, which is about genome. So we just done the discussion about how Segmental sequencing is working, and also this is the sort of the first application in a human genome sequencing, and in earlier times. And we discussed about the the basic terminology about describing the process in a shotgun genome sequencing. So we get the core terminology about the sequencing, like what is the what the shotgun sequencing is, and also we talked about the clone, and we talked about the and also we talked about the reads as well, right? And also yeah, I think that's all. Okay, so that was proposed in 70s, and people try to improve this technology. So improvement means that they want to read more efficient way. So efficient means that it has to be easier and has to be less labor intensive, and also give you better, higher amount of the sequencing product, right? So if you, I mean, a few years later, the scientist Leroy Hood, he came up with the idea about the automated this process. So he basically made a machine and automated all of the process from the preparing the plasmid to the reading the sequence by Segmental, right? So the read length is still around like less than 1,000 base pair, but I mean it's way less the energies and labors, and people easily operate this machine for the high amount of the sequences. Okay, so Segmental sequencing is obviously improved over the year, right? So it's first proposed in 70s. At that time, one hard worker, working technician, he can look at 700 base pair per day. So I mean, sure, he is working on this Segmental sequencing and do the preparation without eating, without sleeping, then you can read 700 base pair per day, right? So if you want to read the entire sequence of the human genome, it takes about 100,000 years, right? So it's quite long days. And after a few years, Littlewood came up with the idea about the automation, and he basically automated all of this process in a sequencing machine, but still 5,000 base pair per day, and it still takes like 10,000 years, right? And after a year, it improved the machine. So ABI system, developed by Littlewood, it's better throughput and technology, and it still, the few base pairs, but takes like 4,000 years. And also after the years, still, 400,000 base pair per day, but still takes, 200 years to, sequence entire human genome, right? So how people did that for the, human genome project is they basically looking at all of these, the entire genome by, lots of people, lots of machine, and also, of course, the money, and also lots of labors as well. So as I mentioned, the two bodies they working in this, human genome project. So first one is the public sectors by, the U.S. government, NIH, and the other one is private company operated by Greg Venter, the Cellular Corporation. So they basically put lots of money onto that. So NIH, they invest like $3 billion onto this human genome project, and Cellular also provides lots of investment, like $300 million, and they try to finish the human genome sequence, and they basically compete this situation. But the strategy is still different. So NIH way is do the hierarchical shotgun. So hierarchical means that they basically chop up the human genome into the large piece at the first time. So this is the entire genome, and they did the large fragment, the fragmentation, like 4 pieces on here. And once you get the large fragment, then you fragment it again. So you fragment it into much smaller pieces, right? So if you get 4 pieces on here and take one large chunk and put it into tens of pieces into the one. And why did that in this hierarchical shotgun sequencing? Because DNA sequences lots of redundant characters. So if you look at the DNA, it's obviously consists of ATGC. So this is, we could say that less complexity in this sequence. If you have the 20 alphabets, then you can easily build up, think about the origins of the sequence, but this is only the 4 combination of sequence. So if you get lots of A or lots of G or lots of C, we say these patterns are repeats. So if you have sort of these repeats, then you're having a problem in the mapping the original position. So they've done basically the hierarchical shotgun and put it into the make a puzzle out of that, right? So they basically building up the entire sequence, construct the entire sequence from the lots of contigs, right? And then different ways is the Cellular Corporation. They basically done the whole genome shotgun, so they don't make any large fragmentation from the genome. They basically chop up the same length, smaller pieces, and they put it into the entire the assembly from these lots of pieces, right? So if you do this way, you probably use much more computation to build up each possibility, because this is much a large number of pieces, and also a large number of possibility into the assembly situation. So I would say 2 different bodies, they use 2 different techniques, but end up in a quite the similar timeline. Okay, so back to the public sector's way. So Human Genome Project, as I mentioned, that this is based on the hierarchical shotgun sequencing. And at the first time, they did the 60,000 large insert clones. then they chop up into the much smaller pieces, and after that, they assemble all of these pieces into the sequence, then they looking at the entire human genome sequence. And in this process, they use lots of different cloning ways. So they use the bacterial artificial chromosomes or P1 artificial chromosomes or yeast artificial chromosomes. So if you do the cloning of your smaller sequence piece, then you can use basically all of different cloning ways to make more DNA reads from the sequence. But I mean, you don't have to memorize all of things. This is just how people use it at the time, and no one really use this cloning technology anymore. So this is, we can say, I mean, the information from the archive, right? Okay, but the important thing is that, as I mentioned, that this DNA sequence is quite redundant. Redundant means that less complexity, and they are repeated by the small number of alphabets, right? A, T, G, C. So this is really hard to make, the assembly, make a combination to infer the original sequence, right? So that's why, I mean, at the time, people came up with, developing of different algorithm, and they tried to do the accurate assembly from these small sequences. So at the time, people used the terminology called quantic. So this is like, I mean, especially for Korean students, I mean, if you see the drama, or if you see, movie, the movie Star Wars drama, the actors, they say conti, right? So this is basically the same way. So you have the small pieces of information from the shotgun sequencing, and you use this, the quantic, and create the entire sequence, right? So if you have, series of quantic, then you ultimately you assemble all of these quantic, then you can make an entire human genome sequence. So how you do the assembly from this quantic sequence, you basically use the overlapping area, right? So if you have lots of, the reads from the cloning, then you're looking at the overlapping sequence on here, right? Then you can easily infer that, okay, so these two reads probably from the same area, right? So then you can infer this piece of information, right? But again, this overlapping, finding the overlapping area is also depends on how redundant the sequence in this overlapping area, right? So if you use only 2 base pairs, so you're not gonna have the assembly of the quantics, right? Because 2 base pairs could be only 4 to 2 combinations, so it's only 16 different combinations. So you have to come up with something more complex than just a few base pairs, right? So overlapping sequences has to be some chunk of the DNA sequence, and you have to mapping those areas, right? So the quantic is the core term, how you build up, how you assemble your sequence from the shotgun situation. Okay, so after all of this, the complicated time, people, they finished the first version of the human genome. So that was happening at the 2001. So this is Human Genome Project Consortium. So HGP is the Human Genome Project. They finished the first draft on the 2001, and 2001. I mean, definitely the very initial version of the human genome. So they try to improve their the quality of the human genome sequence, right? So basically, people try to put their sort of high quality reference sequence making. So they've done lots of job during the the time between the 2000 and 2004. So in 2004, they published a near complete sequence. I mean, that time people said that was the really great improvement in making the reference genome. So what it contains like 99% of the eukaryotic genome, and it only interrupted by 300 gaps. So gaps means that the missing area from the chromosomal positions, right? And also they've done lots of the high fidelity. So high fidelity means the high accurate sequence from the reference genome making. So that means that they only allowed one nucleotide error per 100,000 base pairs, right? So it's only 0.001% error rate from the sequencing. Okay, so but still, lots of gaps in our genome, and that is the sort of problem. We're not going to get the really good, the entire picture of the genome. People still working on, and still people updating the reference genome as we update, the iOS version or Android version, something. So, improvement is still going on. So, where does, these improvements has been happen? It's mainly happening in the repetitive regions. So if you look at the genomic position, lots of positions has only, whatever the G or T or C or A. So this area is still, we're not going to have really reliable assembly from that area. And also we have about, 200 megabase pairs of heterochromatic sequence, which is not really unwind from the cell. So this is really hard to access to the sequence. And also this area is mostly, happening around the centromere area or the short arms of the acrocentric chromosomes. So if you look at the acrocentric chromosomes, you see the P arm. Those areas is still, the making, the genome is still going on. Okay, so from the complete version of the human genome sequencing, what we know about the number of protein coding genes. So that is really, a significant finding. And also people are quite surprised by the, the large number of the segmental duplication in our genome. So, we didn't know anything about our genome. So, people thought that our genome is quite unique, but it actually not. Our genome has lots of the duplication. So duplication means that the DNA sequence is quite identical between the region, between the genomic region, because during the evolution, we have the genomic change throughout the gene duplication. So especially if you look at the primate, we have lots of rapid expansion of our our genomic position by the gene duplication. So we, if you look at the primate, if you look at the humans, we have much more complicated the functions, like higher level functions like cognitions or emotions. The reason is that if you look at the gene, we have much more complexity, we have much more diverse gene composition. So if you look at the synapse is the constitute in a brain. So this is called the brain cells or neurons, and these neurons has much more complicated and also operate by the diverse the protein families. The reason is that during the evolution we have much more the types of synaptic molecules or synaptic genes because of the gene duplication. So if you look at the synaptic genes, we only have one type of synaptic genes. We have multiple types of synaptic genes that increase you the capacity in a cognition and emotional function. So if you look at the segmental duplication, we have lots of different composition in our genome, and also we know about how much the vertebrate specific our genomic sequence is compared to other animals, right? Okay, and back to the segmental duplication. So it's still going on for the improvement. So if you look at the chromosome 1 to chromosome 22 and X and Y, you see lots of areas still on the segmental duplication, which is the colored by segmental blue, right? Colored by blue on here, like in this here, right? So it's only shown for the length greater than 10 kilobase, so which is quite high, right? And also large duplication, it's happening on like in here, right? We have lots of them, and also the gaps. Okay, the sequencing gaps around the segmental duplication or within the segmental duplication. So this area we haven't done really good, really high accurate sequencing for the human genome. So this is the area still undergoing the improvement, right? And this is mainly by the segmental duplication as well. Okay, so if you look at the human reference genome, it's updated over the year, right? So from the 2004, 6, 9, and 13, we updated our human genome reference version. So from the version 17 and 2016 was 18, 2009, 19, 2013 was the 38. So this is quite old, right? The reason is that two institutes, they've done the curation of the human genome reference. So UCSC, which is the genome browser we see last time, right? So this is the operated by the UCSC, which is the University of California, Santa Clara, and also another institute operate the human genome, which is the Genome Reference Consortium. This is the consortium of the human genome reference. So they call the human genome 17 version is 35, right? And then after that, 36.1 and 37. And at the point and here, 2009 is sort of the area, the era people intensively studied the genomic research, and at the time, people felt that why you guys have all different the naming, because they basically use the same genomic sequence, but their notation is slightly different. But other than that, it's nearly identical. So people quite confused about the situation. So they unify their notation from HG38 and also GRC HG38, right? So this point people call the 38 version. So if you go to database of the genomic sequence, then you click the this is the one of the database called GenCode. Then if you go to here, then if you look at the older release version, people still updating their versions over the time, right? So from here, this is from the 36, 37, 38, and within this major version, people has the not small, but some not really major, but like a minor updates for that, right? So this is now currently we have the 38 version of the 38, right? So this is another things you have to keep in mind. And why we update that? Because we have lots of gaps, and sometimes the IDs or the positions are wrong, so people wants to fix that. So this is the how we update the version. Okay, so next part is the initial impact of the Human Genome Project. So if you go to this link, this is the I basically referenced the review by Eric Renda in 2011. This is the paper called the initial impact of the sequencing of the human genome, right? So this is like textbook for your learning in this section, right? So if you read that, how people summarize the Human Genome Project at the time, I obviously summarized a few key points for you. So Eric Renda is one of the greatest scientists in these days, and if you are quite keen on the politics, you probably see that the new president in U.S., the Biden government, they appointed Eric Renda for the director of the science policy, right? So quite amazing move, right? So I mean, Eric Lander is obviously one of the greatest, geneticists in our history. Like, he is probably the modern Gregor Mendel, and he's, he done lots of great research, and also lots of great findings as well. And he's now for the science policy, which is, quite jealous. I mean, given our situation. So what he summarized at the time. So with the Human Genome Project, we definitely, boost our effort in genome sequencing. So I mean, the one key message is we definitely finishing the human genome reference. So that's the good thing. And with this success, people migrate into the different, the genome, different animals or plants. So they start to making reference genome sequence for all, lots of, animals and plants, like, the cute plants, cute animals, I mean, ugly animals, all kinds of animals. They done lots of things. And now we have, comprehensive version of the wide range of, different vertebrates, genomes, and mouse, dog, rat, chimps, cows, as well as marsupials, monotremes, and birds. And I mean, if you really are a fan of certain animals, then you can go to Genome Browser and see their sequence. It's amazing, right? So that's a great thing. And lots of great things is coming. And like the PET 1000 genome projects, I really surprised by this, because PET is amazing animals. This is between the birds and the mammals, and how we see their the meanings in an evolution, and they definitely have their unique the perspective in terms of the agings or navigation, ecosystem, like communication or limbic development as well. So if you know about their genomic sequence, then we can see lots of insights from the the human the animal evolution. And also if you go to UK done lots of great things for science, and they for their 25th anniversary of the Wellcome Sanger Institute, which is one of the greatest genetics institute in the world, and they celebrate their 25 years anniversary. And what they've done at the time, what they plan to do at the time, was they look at the 25 genomes in under the the danger of extinction, right? So I mean, this is amazing things. Like if you think about like Korean Institute, like some anniversary time, they they do the the photo time, and they raise the hands or whatever, the hard things by hands, the stupid things. But I mean, the UK done some productive things rather than just taking photos, right? So they start doing some the genome sequencing for the sort of widely, the endangered species. So, I mean, this is definitely we need to learn from them. Okay, and also, needless to say, if you look at the, the COVID-19 situation, we see great advancement of the genome sequencing technology, right? So, this is the article for the timeline of the COVID-19, right? So COVID-19 was observed in the last day of the 2019, and it's reported by the WHO at the time, right? And after, only within 2 weeks, they done the first draft genome of the COVID-19 by genome sequencing technology. This is amazing things, because if you think about 20 years ago, even like 10 years ago, we have the let's imagine the situation, we get the new virus happening in pandemic in somewhere, and people don't know anything about what to do in their research. But the situation is totally changing now, right? So COVID-19 outbreak at the time, and within few days, they've sequenced the the genome information from the COVID-19, and they embark on the developing of the vaccine from this sequence, right? That only happened like 4 months, right? So biology is is amazingly, has been improved over the time. Who predict this situation in 10 or 20 years ago, right? So I mean, without the the development of the genome technology, we're not going to do these things anymore, right? So I mean, I don't have to emphasize anymore, like genomic sequencing is nowadays is like a routine technology in our daily life, right? So that is the sort of major things we can think about in a human genome project. And the second is we well know about the genome anatomy and physiology, right? So before the human genome project, which is before the 2001, when we're talking about how many protein coding genes in our genome, and people say probably 40,000 protein coding genes, or probably 10,000 protein coding genes, or 10,000 or something, because people came up with all different numbers because we don't have the entire pictures of the human genome project. But after the human genome project, now people know about the exact number of the human genome project. So now we know about the protein coding genes. We have around 20,000 genes in our genome, right? So this is definitely one of the great advancement by the HGP. And also, most important thing is we know about most of our genome is constituted by non-coding genome sequence. So if you look at the area that can produce the protein coding sequence, this is only about 1.5% of the genome, right? So out of 3 billion base pairs in our genome, we only have about 1%. The sequence can provide you the proteins constitute your body, right? Other 99% is actually the non-coding sequence, which is not going to process any protein sequence. And then people was curious about what those reasons do in your biology. So the main hypothesis at the time was they probably do the gene regulation. They probably do something important for your development, right? So they probably do some functional things in other than just protein, other than just the proteins, right? So people came up with the idea about how these areas are regulated, how these non-coding regions are regulating our bodies and our development. So one way is to conserve the non-coding element, which is the conserved across the species. So not just in the human, not just in primate, it's happening in a mammals, or it's happening in a vertebrate. And those regions are like, protein coding genes. They are actually working in a certain biological pathways, biological process that can, make sure the proper entity during your development. And these elements also we can identify from the human genome project, right? And this is probably TMI, but when I was in students, I was at the time, and these non-coding regions are finding at the time. So we are quite excited by the latest paper at the time then, that research is like microRNAs or non-coding RNAs, right? So like you, when I was student, the papers about microRNAs or non-coding RNAs, they come up in a Nature or Cells, and like you guys, when I in my class, my lectures, they gave us the papers for the microRNAs or non-coding RNAs, and we read that paper for the assignment as well. And that's like 15 years ago, right? So and the field was really the mature over the time, and nowadays is well appreciate in a science field, right? And also some researchers like they really well known in our country as well. So you probably see the news about the professor Kim Bin-neri last weekend. She appointed as a fellows of Royal Society in Britain, right? So this is like, only 300 scientists over the 200 years, like, Isaac Newton's or Charles Darwin. So she's one of the fellow at the time. She is, dedicated her research into the microRNA, the biology, and I mean, everyone in the world know about her research, right? So, this is obviously the starting point was the, knowing the human genome sequencing information at the time, and people having a, the thought about how those non-coding regions can be functional during the development, right? Okay, and obviously these types of research is still going on, and also still, quite fascinating the topics in our field. Okay, and also another, big point is that we know about the variation of our alleles in a population, right? So we have a long, discussion from the March to today about, how we understand sort of the particles proposed by Gregor Mendel 100 years ago, and how these things related to the alleles discussed in these days. And this is the one of the cornerstones we can discuss this idea is the Human Genome Project, and we know about the sequence, and based on this sequence, now we can accurately measure the frequency of the allele and the positions of the alleles, right? And because of the Human Genome Project, lots of consecutive efforts happening in human genetics, like in HAMMET project or SNP microarray, and also we now better understanding, regarding the copy number variants or the structure variation we discussed in session 3, right? So all of these things is all sourced from the Human Genome Project, and that means again you have to read the Lender's paper 2011, and also you might find interesting if you compare the situation that time and nowadays, right? Because it's about 10 years differences. We probably have better the advancement in these days, right? Okay, that's all good. And this is the same, the review by Ireland at the time. And also I put the, his lecture. Basically he done lots of lectures, and you can find from the YouTube, and I just copied one of them. And this is the Killian lecture in 2017. And he's a great scientist, but he's a great lecturer as well. So, in MIT, he teach human genetics. I just laugh because, I felt a bit, pathetic myself because, compared to him, but Korean university, but a different situation. But anyway, so if you go to YouTube, then you can get his amazing lecture. So this is one of his. So I hope you enjoy this lecture as well. Okay, so go to session 4 and 2. Oh yeah, so I sometimes feel a bit. I sometimes feel a bit. Doesn't make sense about the the university lecture in our system, because sometimes I feel lecture by presentation killing your creativity, killing your the thinking ability. The reason is that the presentation cannot capture entire contents into the into the sections, right? So presentation is very limited space for the information, right? So, but we are, we've been tamed for reading presentation, even in university time too. So I don't know whether this is the legit or not, because some of the lectures I really like for science, especially science topic. It's actually based on the the board, right? So it's like explained by it's like, you know, chop talk, but these days, you know, we open up the presentation, we read the presentation things, and I mean, just deliver sort of the piecemeal information of the entire science. So I mean, I don't know whether this is really good for the, you know, the next generation, but you know, what can I do? So so what I can recommend at least is, you know, go to, you know, the internet browser and search some, you know, the deep information from that and build up your thought out of that. But one point I can help you is, you know, sort of the curating the good research for your reading and education. So you know, Linda's paper and lectures is one of that, right? So don't limit your capacity to learn more things, not, I mean, from this stupid presentation situation, right? So, okay, so go to session 4 and 2. So be more positive on the learning. So genome sequencing. So this is the repeat of the Senya sequencing part. But before we're moving on to the next session, so what is the genome sequencing? So I mean, this is the process of determining the nucleic acid sequence, right? So the order of the DNA nucleotides in your genome, right? So basically, reading the sequence is to determine the genotypes. This is obvious, right? So we know about the genomic sequence because of the Human Genome Project and because of the next generation sequencing, right? So now we know about the sort of the Mendelian particles he proposed at the time, right? So nowadays we call the variant or we call the genotypes, right? And again, this is the allele at a locus. So allele at a locus means that you know about the composition of the DNA sequence, but also the positions, right? So you have the two layers of the information, right? Okay, and also we have to know about the allele, right? So that means the changes in the nucleotide sequence compared to reference genome. So we call the variant, right? So genome sequencing is basically the basic information finding the link between the genotype and phenotype, right? Okay, so what should I show you first? Okay, so genome sequencing. Okay, so I probably want to show you this first. So this is the UCSC genome browser. So so we now know about the reference genome sequence, right? So we can definitely see the sequence of certain gene from the genomic browser, right? So genome browser, right? So if you go to UCSC genome browser, so search from the Google, then you can easily find the link. Then if you go to the gene, I mean, SN2A, this is the examples of our class, right? So SN2A is obviously has the gene information, right? So if you zoom out, it's very fast. Zoom out again. Zoom out again. Cool. Okay, so this is the SN2A position. So that's on the chromosome 2 and the Q arm 24.3, and that's near the other genes, right? So SN3A, so SN2A. I mean, they look similar, right? So this is the sodium channel, and this is the sodium channel. So we call that they from the same gene family. But not always the genes from the same family, they located near each other, right? So this is a different, right? So SN2A, but like other SN something A, they can be located on the chromosome 5, they can be located on the chromosome 8 or something, right? So there is no rule for that. They are spread out across the chromosomes. But certain situation, the genes from the same gene family, they locate, in serial position. In this situation, we call the synteny, right? So I didn't put this specifically into here. So this is called the synteny, right? So during the gene duplication, sometimes the duplication happen near each other in a near position. So that case we call the synteny, right? But synteny is sometimes happen in a certain gene family, sometimes not, right? So I mean, we kind of make a generalization, but I mean, it's certainly happening. Okay. So SN2A, so it has the start position here and stop position here and this direction, right? So this arrow means the direction. So the transcription happening from here to here, right? 5' to 3', right? But SN3A is different, right? So transcription start from here to here because the direction is this way, right? So this is also from the 5' to 3', but that is located on the reverse sequence, right? So that's why it has the different direction. Okay, so where we have to go, we want to find start codon. We want to find, oh, great. Okay, so yeah, no. Okay, we can zoom in. Then we can zoom in again because we're not gonna, we cannot see the sequence from here, right? But if you can zoom in, then we can see the DNA sequence information here, right? So if you read here, GATG, GATG, right? Then you will find start codon position, right? ATG, methionine here, right? So the translation start from here, right? And you can obviously see the sequence information. Then you can also see whether this sequence is conserved in other species. So if you look at your lovely dog sequence, it's well conserved, right? So it's exactly saying, you know, sequence as human in this position, right? But unfortunately, your lovely zebrafish doesn't have the two missing codons on here, right? So this is not really conserved. Okay, so this is the one way we can look at the reference genome. Another way is you can download okay. Oh no. Okay, so I mean, if you find really, you know, interesting, you know, looking at the DNA sequence, then you can go to GenCode. Then you can go to here, the current release. Then you can download the gene position, download in here, or you can download the entire sequence from the FASTA file, right? So I mean, we're not gonna go to very detailed today, but sometime, right? So if you click here, then you can download the file as well. So how does it look like? It's look like it's this. Okay, so ready to see. Okay, so if you look at this is the human genome reference sequence. It's amazing, right? So this is the chromosome 1, and this is the first position. So the human genome, the first position is N. It's not ATGC. So why it's N? The reason is the N means that we don't know the sequence. We don't know this area. So just mask that area by N character. So why we don't know about that area? The reason is that this area is the telomere, right? So telomere or the centromere, all of this area is still hard to sequence. So we don't have the information. But if you go scroll down, I'm actually scrolling down, then you can see the sequence information like this, right? So you see TAA, CTT, whatever, blah, blah, blah. So if you want to find the start position, then you can type ATG. But obviously, this is not going to give you the start position. There must be something rule for that because it has to be codon position, right? So okay, so CHR1, so chromosome 1, but there is also chromosome 2, right? I mean, it takes some time to search the chromosome 2 because so it's chromosome 2. So do you remember where the start position of the SN2A start codon? There was this, right? So there was the 165,295,824, right? So if you want to find the area, then you can you can count this, but no one gonna do that, right? So don't do that, right? So you're not gonna take class next week, so just quit. And there's a way we can do this. We can find the ATG position by programming like Python or R. R doesn't really great on that, but you can use Python. Okay, or we know about the genomic position, about the genes or transcript. Then you can look at the GTF file, which is the first file I indicated at the website. So if you go to here, then you can see chromosome position and the gene and gene ID and the gene name DDX11L1, right? So this is the first gene in our chromosomes, right? Chromosome 1. Then SN2A, we can also find that, like in here, take some time. Okay, cool. So chromosome 2, gene, this position, and gene name, blah, 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 right? So obviously you're not gonna search by your hands because, we are living in a, good time, so computer do that. So how to operate this search by computer, biostatistics next semester. Amazing class. So back to the Brave browser. Okay, so moving on to the next generation sequencing. Okay, so we have the genome sequencing at the previous generation, which is the Sanger sequencing, right? And after Sanger sequencing, we came up with the better technology called next generation sequencing, right? So next generation sequencing came in 2005. The first machine by Roche, 454, it's gone. So I'm not gonna memorize these things, right? So not important. In 2006, the new machine from different company called Solexa. And this Solexa merged by other company called Illumina. And nowadays, you have to know this company. This is like, equivalent to Apple, right? So, I mean, people will laugh at, Illumina like Apple, people will be laugh, but Illumina is actually really big company, right? So this is one of the, the renovated company at the first time, but nowadays they dominate the market. So these days, everyone uses the Illumina, right? So Illumina takes the merge, the Solexa technology, and they spin up their new machine after that, right? So I was quite lucky because I learned the technology from the Illumina at the time in 2010. Then I learned about the machine from the Illumina. Then I could be on a Illumina company, but I took the different career from that. But anyway, so Illumina was one of the renovated company at the time. So and after that, lots of different companies, they generate sort of the competitors, right? So ABI, which is the same, the ABI system on here, right? And they, with the solid and by the live technology, this is another, the big company, and they came up with the machine at the time. ABI solid system was good, but this is also obsolete. So no one gonna use anymore. Complete genomics came up in 2010. Complete genomics is okay at the time, but this is also obsolete again. And Pacific Bio, this is the came up in 2011. They are still active in the market, and they're quite good, the machine and technology, but very expensive. So they're not dominating the market. So if you think about the situation, like the iPhone or Galaxy or Xiaomi or something. So sequencing technology is basically same. So Illumina machine is like an Apple, or Pacific Buyers is like something. So they are competing the price, they are competing the speed, they are competing the capacity, they're competing the the tech, right? So a spec, right? So Pacific Buyers is good technology but still developing. Ion Torrent is also obsolete. I mean, I would say it's obsolete technology, but not many people use the Ion Torrent. I don't know. I might be sued by Ion Torrent, but I should say, this is not. I'm not my technology, but somehow in Korea, some hospital or some companies, they bought lots of Ion Torrent. I don't know why, but I would run away from the Ion Torrent if heard that your organization use Ion Torrent. I'll tell you why. And Oxford Nanotechnology, shortly ONT. This is one of the renovated technology as well. Lots of people use these days, but different perspective, right? So why we have all of these different technology and companies for the genome sequencing, the reason is that they want to generate more data. They want to generate more DNA sequence, and they want to generate that information in a quicker way, right? So they sometimes call it massively parallel sequencing because they put the sequencing process into the parallel situation, not just the single workflow, right? So put it into the parallel way. And also they called it high throughput. So you probably get these words many times. This is one of the key words in this session. So throughput means that the amount of the data set, right? So throughput means that the size of the data set you can generate from the sequencing run, right? So literally high throughput means that it gives you large amount of data, right? So think about this situation. If you use the senior sequencing technology 77 version, you only get the 700 base pair per day, right? But if you use the Illumina sequencing technology these days in 2021, you get 1 petabyte base pairs, right? So this is amazing data size compared to 77, right? So that's why we call the high throughput. And sometimes people call it ultra high throughput because this is like your technology is high throughput, my technology is ultra high throughput, my technology is mega ultra high throughput. So they always fighting in these ways, right? So so some people call it this way, or some people call it the second generation. So first generation is the senior, and the second generation is the next generation sequencing. Okay, so a few principles in next generation sequencing. So next generation sequencing produced sequence reads, right? So this is the same read. So this is the fragments of the nucleotide. And NGS is mostly produced short reads. So fragment your DNA into the very short pieces, like 300 base pairs or 500 base pairs. So this is quite short DNA sequence compared to entire human genome. And after you fragment, then you put it into the library situation. So library is the entity you can put it into the sequencing machine. So once you fragment your DNA, you have to convert that DNA into the format you can do sequencing, right? So that we call them library, right? And sequencing by synthesis, so you add up the LTGC information into there. And also the read length is really depends on the technology. So if you look at the different technology, they have different length of the DNA sequencing. So Illumina at the first time, they only sequence 50 base pair, but nowadays they sequence like 500 base pair. So the length is different. But ONT, they sequence like a 10 kilobase, so 10,000 base pairs. So the read length is really depends on the technology. Okay, so few the terms describing the NGS. One is the throughput, so this is the amount of the output. So one important terminology. Another important terminology is the depth and coverage, right? So depth is how many times the region is sequenced by the reads. So the region means that if you think about the allele, so if you think about the position, let's say, if you want to look at the mutation on this position, right? So this is the position of the 82.5, right? And you want to ask how many times the Illumina sequence reads read this position, right? So of course you can read only one time, but that is the less confidence, right? So you read like 50 times, you read 100 times, or you read sometimes 10,000 times, right? So why you care about the confidence? Because if you think about the situation, you want to diagnose the patients who might be cancer or not. So you want to diagnose by the mutation of certain, let's say lung cancer. Then you want to see the mutation position, let's say in this position. And if you find the mutation from here, then you can diagnose, okay, so this patient is the lung cancer, right? So if you read one time, or if you read 10 times, or if you read the 100 times, of course, if you read 100 times, you can give you more accurate mutation information to this patient, right? So if you go to clinic, like the big hospital, they do the NGS screening, and they do like 1,000 reads or 10,000 reads, because they want to give the more accurate and more confident information to the patient, right? So we care about the depth, right? And another terminology is the coverage. So coverage is how much the region is covered by sequence read. So think about the situation like SN2A. If you want to read this region by Illumina sequencing, then as I mentioned, that in early times, Illumina sequencing only do 50,000 base pairs. So only this part, right? So if you read entire 3,000 base pairs, then you can basically divide up to like 600 different regions, right? So sometimes the reads can be mostly happening in here or mostly happening here by the the technical artifact, right? So you are caring about how uniform the coverage cover that how uniformly the reads cover the region, right? So reads has to be cover all of the region without the missing part, right? So coverage is the terminology describing this that situation. Okay, we run out of time. It's very sad. Okay, so so first technology by the Roche 454 was the Pyro Sequencing. The point is that they make the fragmentation. I mean, the shear means that you chop up the DNA and you add up the adapters at the end of the fragment. So this is the DNA sequence. Then you add up the adapters, which is the all common across the fragment, right? Then what this adapter is doing, this adapters is binds to the beads, right? So this is like a little glove, and this on the little glove, it has the probes binds to these adapters, right? So they basically baiting the DNA fragment with adapters to the beads, and you read the sequence out of these positions. But basically the idea about this adding the adapters or it binds to the probes is basically same across the Illumina or ion torrent sequences. This is basically the same core concept in NGS, but the difference is that they are processing all of this process into the really tiny microfluidic system. So they put it into all of these beads into the wells. So wells means the little little holes like in here. So they put in the beads onto these parts, and they sequence the information from this area, right? So because of this microfluidic system, which is really tiny chip, and they put the tiny beads onto these tiny chips, and they reading the DNA information from this area, right? So Pyro sequencing by the 454, they use the same fluorescent and also the primer information, which is quite similar similarly used in the Segmental sequencing, right? So they use the all different colors of fluorescent for the 4 nucleotides, and they synthesize those nucleotides into the well system, and they read out the fluorescent information onto the technology, right? So Pyro sequencing, I think is, and also they automate that system into the machine as well. So they read out the information from there. So they've done the James Watson's genome at the first time in 2007, and only cost like $2 million at the time. And this is the, after the Segmental sequencing, that is the first NGS technology that can easily and they can quickly read out the genomic information from person's genome, right? So people really excited about the possibility of NGS technology in a human genome area, and people moving on to the technology and people rapidly working on the situation. But again, 454 is the technology gone these days, so it's rapidly the outpaced by other technology like in Illumina. So if you think about the NGS technology, you probably keep the two technology, which is the first one is Illumina technology, and the second one is the long read sequencing by nanopore technology, which are introduced on here. Okay, so run out of time, so I back to from this part, Illumina sequencing technology. It has lots of the graphic information about how technology is working, right? So, and also, as I mentioned, the Illumina is the technology dominate the market. So, lots of the YouTube video clip for the technology. So you probably go to some of the link, then you can see how the technology is working. But we're going to review this one in next week as well, right? And then we move on to the long read technology. Okay, I think that's all. Okay, so we back to here. Okay, and also another the short announcement is that our what is that? The our Wednesday class is the we have no class for this Wednesday because I have another schedule, so I'm really sorry for that. So I probably miss the this week class, or I probably upload the replacement of the today's topic onto the YouTube. Then you can follow up from there. So I announced once it's decided. Okay, so I think that's all good, and we resume our session from here next Monday. And I hope you enjoy the rest of the week. And again, I mean, as always, if you have any question or feedbacks, I mean, it's always welcome if you leave yours onto the Slack channel. Okay, so see you next Monday.